Excellent. So it's time for our question and answer time. I do have a question for you, Yarmali. I find the microsampling techniques very interesting. And I remember that you showed me in person in the lab how the microsampling wing works and the VAMs and all of that. I wonder, I know it's very challenging. I wonder, how do you deal with unstable drugs?
So if it is an unstable drug, you need to ensure to use the appropriate microsample device, or microsampling device. For example, if it is in the case of cefotaxime and that is not stable in the right format, that's why we chose the capillary tubes to be able to collect the sample and then immediately after centrifugation, just store the sample at the appropriate temperature. That's why in the lab, you have to run several assays or experiments to evaluate the stability of the drug across different times. So vancomycin, for example, is very stable at room temperature in the dry format. So that's why vancomycin is a really good drug to do that. And Traco, and there are other antimicrobials that you can do that. But once you know that the drug is not stable, so you definitely need to move to another format.
Thanks, Yamali. I've got a question for you, and then Abby, I have a question for you. For Yamali, I imagine that at this stage, the analytical approach to assay is based on chromatography, which is for some antimicrobials, not as convenient as the immunoassays that currently exist for the aminoglycosides and glycopeptides or vancomycin at the moment. I was wondering whether you could comment on the prospect of developing immunoassays for these microsamples, and it may just be one of those things which just requires a significant body of work, and so is a bit uncertain, just to help you out with an answer if you're unsure. Or alternatively, it may be just because of the sample volume, it's very difficult to conceive how this could be solved by anything else other than chromatography. So this is all about feasibility and being able to roll it out into a clinical laboratory scenario. So I'd be interested in your thoughts about what the implications of microsampling is for a pathology laboratory.
Well, that's a very interesting question. I'm not quite familiar with these drugs, and I think we are all familiar as well with, for example, Theranos and the situation that's just trying to use as one drop of blood and assaying several laboratory tests. So I think the technology in that perspective, it's more complicated than we think. Mass spectrometry, it's an incredible technology that we can assay several drugs, but it doesn't work quite well to every single drug. And in terms of the pathology, I believe it has to be something more no, I would say complicated, and it's different from this one. But there are other sample and drugs that can be easily, I say, as in the pathology as well, using mass spectrometry, and they can be applied to this one. The thing is, they might take longer than in the safe compared to, for example, antimicrobials that sometimes for us is just a 5 minutes run or 10-minute run, but some molecules take a longer to assay.
Thanks very much, Yamali. I'm actually going to ask Abby the same question, although of course the immunosuppressants are typically assayed by chromatographic methods, but what she thinks about that whole prospect of using different bioanalytical approaches, such as immunoassays, for these compounds, which may actually increase the feasibility of microsampling to be used at other laboratories as opposed to major pathology laboratories.
So I think there are all of the issues associated with like the immunoassays in general in transplantation. The newer ones are much better as far as, you know, the sensitivity and specificity go, where we're not quite as worried as we were originally about the metabolite contribution and increasing the drug concentration. But we still are very particular about our assays. Sorry, the cat was being annoying. I had to open the door. So even here at the University of Michigan, even today, with the quality of the assays, patients will mail us their whole blood sample to run their tacrolimus concentration here rather than at the outside lab that may be closer to their home. And so what I really view this as making a chromatography more feasible. Because you could throw it in the mail without ice, without having to worry about the stability, and have it analyzed all at the same laboratory with chromatography methods. And I'm not even familiar if there is a mycophenolic acid assay that isn't chromatography. So really, I view it as making chromatography more feasible for labs that don't currently have that option.
That's great, thank you. My next question for you, Abby, relates to other analytes that could be looked at in the same sample. So you mentioned serum creatinine as an option. There's been at least one or maybe two papers which have tried to develop an assay, including serum creatinine with tacrolimus measurement in microsamples. But what about other drugs that these patients are on as well? So for example, steroids, there isn't necessarily a particularly strong exposure effect relationship that has been defined for these, but I would expect that something like that must exist. And so that would be a really important area of research to go down to try to really understand that holistic approach to therapeutic optimization. I wonder if you could just comment on that general area for me.
So there is one paper, I think there's only one, that has been published that added I think it was prednisone to the multiplex assay. The problem with that is that very quickly you get past the lower limit of detection, even with chromatography at the doses that we routinely use in transplantation. So I think it is possible, but it would probably be more looking at something like a peak concentration rather than your trough concentration, or being able to estimate a real, a meaningful AUC. Because even though we can't detect the drug, we believe we're still having a pharmacodynamic effect. And so I think that is a whole nother can of worms, is how we could add prednisone into the mix or other corticosteroids in transplantation, because a solid 60% of patients in the United States are on those as well. And I really do think we need to be taking into account the entire regimen that they're on, not just tacrolimus troughs. So definitely something that I will hopefully get into in the future. But there is, I can't think of who the authors are, but there is one paper that did try to add prednisone. And then, as Dr. Downs mentioned, ganciclovir or valganciclovir, just a couple of weeks ago, we started some initial testing with ganciclovir, and I thought I wasn't going to be able to get it multiplexed because it is much more polar than my immunosuppressants, but it actually looks like we might be able to get some pretty good results from that perspective. And I think that, as was discussed, is an important addition as well.
Fantastic. And my final question is going to go to both of you, but because Abby, you've answered the most recent question, I'll put it to you first. Is what do you think about the likelihood of industry engagement to try and increase the feasibility of this? At the moment, it would very much be led by people that have very much an academic mindset, those that are able to take the steps that, for example, you've been able to do to establish the assay methodology in a laboratory and to motivate the local clinical staff to use the technology. But that isn't necessarily something which is going to grow it particularly widely, because there are only so many Dr. Abby Linos that are around in the world. So that's where industry may be very helpful to try and help propagate it further. Now, there may be a couple of steps that are required before we get to that point. What do you think their likely level of interest in this is?
So I think they do have an interest in it. A year ago, 2 years ago, I can't remember, at the was the American College of Clinical Pharmacology. There was actually an entire session where it was people from industry discussing their use of microsampling in their clinical development programs. And so I do think they have an interest in it. I think in general, the industry's motivation is to not need therapeutic drug monitoring once you get out of their studies. So I think from a clinical perspective, they're not super interested in it because they don't want a drug that requires TDM. But from in my clinical trial to get additional information perspective on the PK, I think they are quite interested in it. And I do feel like they're using it like that migraine phase 3 study where they were having patients collect PK during a migraine, that isn't going to happen in clinic. So I do think that they are applying it and they've done some pretty innovative things with it, but from getting it into clinical practice or actually publishing, that their assay development work, I think it would take a lot of pushing from us, the clinical or academic side of things, to convince them to do it. I think maybe more feasibly would be to get the big clinical labs interested in it, right? I don't know where you are, what's your big clinical labs, but here in the States, Quest Diagnostics or CompuNet, those big lab companies. So we could, send dried samples to them and they would be able to analyze it. I think is potentially more feasible first step.
We might need to invite you to Brisbane, Australia, so you can see what our pathology laboratories are like here. I'm going to ask Yamali the same question, but I'll reframe it in the context of the industries that develop either diagnostics or alternatively those that provide consumables for laboratories. So ready assay kits is more so what I was alluding to. And whether or not you think that there is an industry interest in something like this, or whether or not the possibility is that actually the market's just not big enough, the patients are too spread out for there to be enough interest. No, please, no, that's for you, Yamali, please come on.
It's, I think the industry is very conservative somehow, and it's a little bit complicated sometimes to bring a new approach and just let them know that there is other options available. And the diagnostics laboratories are quite small at the moment, and it's growing in interests, and perhaps with so much technology at the moment, so we can see an improvement on this in the near future. But I think somehow now it's just very small. And I think there is a couple here in Brisbane as well that could potentially do this for in the future or right now.
Thanks very much to both of you. I asked that question because I've had about 4 or 5 companies come to me to talk about B-lactam therapeutic drug monitoring technologies, which they can make more easily available than just what a chromatographic method may require from an infrastructure and staffing perspective. And so I do think personally that there could be a bit of interest if people like Dr. Lino, Dr. Parker, Dr. Dr. Downs, and all of the people that are interested in this area, if they can continue to do the work they do, and that will make this technique and therapeutic optimization strategy available to more. So we're going to conclude there. Thank you very much to the presenters. That was super interesting, high quality research that we got to see very early on in its after having been done. And so it was a great pleasure for us to be able to host this. I'd ask for those of you that are online and those that are in the auditorium here to answer this survey and tell us what you think about the presentations. If there's information that you want us to feed back to the presenters to help them, then that's something which would be really grateful to hear. But otherwise, ideas for what we can continue to do for our education program would be gratefully received. I'd refer you all onto the CRE Respond website so that you can keep an eye on other opportunities and initiatives that we are developing. And you can see from this QR code here, a nice link to that website. And I encourage you all to follow us on Twitter, on LinkedIn, as well as, of course, see our YouTube channel to look at not only the presentations that were provided today, but our entire library, which includes well over 100 fantastic presentations. So I think that what we'll do is we'll conclude there. I'll thank everyone for their participation, to Dr. Mitra for her co-chairing, and the presenters. And I wish you all a wonderful day. Thank you very much.